Good morning, student. Today we will discuss about the genetically engineered insulin. This is the major achievement of biotechnical application production of insulin. You know, many people are suffering from diabetes in whole world. Okay, so that time this achievement is very important for human welfare. So, what is insulin? First, we have to understand that what is the normal structure or natural structure of insulin. Please try to understand it. Okay, insulin is composed of three chains. Okay, naturally, try to understand it. First is A chain, B chain. Okay, and one chain is there which is used to combine these two chains. Are you getting my point? A, B, and C. This is the natural form which is present in humans. And about this natural form, this is the proactive or inactive insulin, and it is known as proinsulin. From where insulin is secreted? Beta cells of islets of Langerhans, and these are present in our pancreas. Okay, so beta cells, please try to understand it. So how many chains are there? A, B, and C. C is naturally present, and C is having work that in A and B, actually they are polypeptide chains. So that time they are having sulfur containing amino acid. So the sulfur is making bond here. Okay, so this sulfur contain this bond. When the sulfur bond forms, at that time only this C chain gets to be dissolved, or it gets to be detached from it. Okay, ultimately which structure will form? The structure of A strand, B strand, and in between of it bond is there, sulfur bond. Okay, this is A chain, it is of B chain, and this is the active insulin in case of humans. Okay, this is active insulin. Earlier was known as the proinsulin. Okay, जैसे वो C chain यहाँ से हटा, ये bond बना, it gets to be activated. This is known as active insulin. Okay, so this you know very well, there is a process of protein synthesis. You know very well, as this process is present in eukaryotic cell. Eukaryotic cell में बेटा protein synthesis कैसे होता है? Please, I am just giving you one recap of it. From gene transcription process is there. Then what forms? hnRNA. From hnRNA, because of the splicing, breaking of this RNA, we are having different part, that is of the intron and exon. Only exon can be ligased because exons are the coding sequence. Introns की जरूरत ही नहीं होती है. So this coding sequence ultimately form with the help of two primers. Primer A or primer B means 5' strand to 3' strand, and this is known as the mRNA. And from this mRNA, the protein synthesis takes place. This is the general process which takes place in eukaryotic cell. But we are synthesizing artificial insulin with the help of E. coli, and E. coli is prokaryotic cell. That time the previous process can't be happen. Okay, so that's why one another method is used in case of synthesis of gene A and gene B. So how gene A और gene B बेटा अलग अलग E. coli में synthesize होते हैं. Okay, gene A and gene B. I am telling you here about the gene A. Firstly, what happened? From gene A, mRNA synthesis takes place, but afterwards what happened, that due to the reverse transcription, ultimately we have to put this recombinant DNA inside the E. coli. E. coli में ही तो synthesis होना है. Okay, so what we do, we make cDNA, and this is known as the complementary DNA. Reverse transcription होने से क्या बनता है बेटा? cDNA. Okay, so what we will do here, we just this is the cDNA. cDNA means which code for A gene. Okay, and here is the plasmid. You just apply this AGNA in this plasmid, और फिर इस plasmid को कहाँ डाल देते हैं बेटा? E. coli के अंदर. Now E. coli is having the gene A carry करने वाला plasmid. Okay, ultimately from this E. coli gene A will synthesize, same process will take place in gene B. Okay, so you are having gene A and gene B. Okay, and they are having the sulfur bond, as I told you. So this is the insulin which is extracted or developed by the biotech method. That time the name has given of it as a humulin. Okay, so this humulin is very important because it's just like human insulin. That's why the name is given humulin. Okay, I hope the process is clear. Children, few things are very important about humulin. That is, it is first synthesized or just commercially produced by one American pharmaceutical company, and the name of this company is Ali Lilly. Okay, it has written here in NCERT book Ali Lilly, and date is very important, children. 5 July 1983. Please note down it. 5 July 1983. Clear? It is just basically synthesized for the management of adult onset diabetes. Adult के में जो diabetes होती है उसके लिए बनाई गई थी. Okay, in 1983. Okay, children. Or this sequence of A and B, it I think is totally clear. One more important thing about related with the discovery of humulin, that is of two scientists name. Please note down is Banting and Best. Okay, Banting and Best isolated insulin from dog's pancreas. Okay, Banting and Best isolated insulin from dog's pancreas, and Frederick Sanger, he gave amino acid sequence for insulin. The another scientist is Frederick Sanger, who has given amino acid sequence for insulin. जब मैंने amino acid sequence की study करी, that time it has become very easy to produce artificial insulin. Okay, one more discovery related with the insulin is Ali Lilly company, pharmaceutical company, and Ranbaxy. They both combinedly form homolog. What is homolog? Homolog is just an analog of insulin. So homolog एक बहुत रैपिड प्रोसेस है इंसुलिन को प्रोड्यूस करने की. ओके, सो दिस ह्यूमोलॉग इज वेरी लेस टाइम टेकिंग. इट इज जस्ट लाइक अ ह्यूमुलिन ओनली, or it is just an analog of insulin. Okay, so after the rediscovery, the new things come to be existence. So it's about the insulin. I hope that is clear. If you have any query regarding insulin, please send me. Thank you.